Hello. Tonight we'll spend some time with one of the oldest democracies on Earth, one that has been running without interruption for roughly 100 million years. No parliament buildings, no written constitutions, no elected officials who forget their promises by morning. Just a colony of 50,000 individuals, each one smaller than your thumbnail, collectively making decisions of extraordinary precision and fairness. Scientists who study group behavior have found that bee swarms, when choosing a new home, outperform most human committees on nearly every measurable criterion: speed, accuracy, and the ability to reach consensus without a single voice, dominating the rest. Tonight we'll move through what these creatures are, how they think, how they speak to one another, where they come from, and finally how they rest. Settle in. There is no rush here. The hive has been doing this long before we arrived, and it will carry on long after. All we have to do is listen. A honeybee's eye does not work the way yours does. Where a human eye holds a single lens that gathers and focuses light into one continuous image, a bee's compound eye is built from thousands of individual units called ommatidia, each one a complete optical system pointing in a slightly different direction, feeding a slightly different slice of the world into the brain. A honeybee has roughly 6,000 of these units in each eye. The result is a mosaic image, lower in fine resolution than human vision, but dramatically wider in field and exquisitely sensitive to motion. More striking still, bees see into the ultraviolet spectrum, a range of light completely invisible to the human eye. Flowers that appear plain white or uniformly yellow to us, in ultraviolet painted with bold patterns, landing guides, nectar markers, visual signals that have co-evolved specifically for bee perception. The bee does not see a meadow the way you see a meadow. It sees a lit runway, every flower broadcasting its own arrival signal in a frequency we will never access without instruments. Every honeybee is built on the same architectural blueprint that has served insects for hundreds of millions of years. 3 body segments, 6 legs, and a pair of antennae. The head carries the sensory equipment, eyes, antennae, and mouthparts. The thorax, the middle segment, is almost entirely muscle and acts as the locomotion center, anchoring all 6 legs and both pairs of wings. The abdomen contains the digestive system, reproductive organs, and in workers the wax glands and the stinger. What makes this plan remarkable is not its uniqueness, many insects share it, but the extraordinary specialization layered onto each component across millions of years of evolution. The legs of a worker bee are not generic insect legs. They carry pollen combs, pollen presses, and the corbiculae, the pollen baskets, each one a purpose-built structure that would require significant engineering to replicate in any material other than chitin and hair. The body is compact. Everything has a job. Nothing is decorative. A honeybee has 4 wings, not 2. They are arranged in 2 pairs, a larger forewing and a smaller hind wing on each side. And during flight, they lock together along a row of tiny hooks called hamuli, functioning as a single unified surface. At rest, they separate and fold neatly to the sides of the body. In flight, they beat at roughly 230 strokes per second, a frequency so high it produces the familiar hum that has accompanied human outdoor life for as long as humans have been outdoors. That sound is not incidental. It is the audible signature of a mechanical system under considerable load. The wings themselves are not moved by direct muscle attachment, as in many other insects, but by indirect flight muscles that deform the thorax itself, causing the wings to follow. This indirect system allows the extraordinarily high beat frequency that would be impossible if each stroke required a separate muscle contraction. The engineering is elegant. The sound is unmistakable. Most people recognize it before they consciously register what made it. The thorax of a honeybee is by proportion one of the most powerful structures in the animal kingdom. It occupies a significant fraction of the bee's total body volume and is packed almost entirely with the indirect flight muscles that drive the wings. These same muscles do double duty. They are also the heat engine of the hive. Before a bee takes flight in cold conditions, it decouples its wings and shivers these muscles, rapidly warming its body to flight temperature without moving from the spot. On cold mornings, foragers can be seen doing exactly this on the landing board, vibrating in place, building internal temperature like a pilot running an engine check before takeoff. The thorax is also where the waggle dance is powered. The vigorous, full-body vibration that a scout bee performs to communicate direction and distance requires sustained muscular effort. A bee, advertising a particularly excellent food source, will dance for minutes at a time, burning energy to broadcast information, a physiological investment in collective knowledge. A honeybee in full foraging trim weighs somewhere around 80 milligrams, about the same as a single raindrop on the heavier end of the scale. When it returns from a successful foraging run, it may be carrying a nectar load that brings it close to 160 milligrams total, effectively doubling its body weight. That is the rough equivalent of a person stepping onto an aeroplane carrying another adult human being on their back using only muscle power to get airborne. For many years, aerodynamicists could not fully explain how bees generated enough lift for this. Early fixed-wing calculations, which assumed smooth gliding flight, suggested it should be impossible. High-speed photography eventually revealed the answer: bee wings do not glide. They rotate at the tip on each stroke, generating small, fast-moving vortices of air that produce lift through a completely different mechanism than aircraft wings. The bee was not defying physics. The physics was simply more complex than the models assumed. It usually is. On each of a worker bee's hind legs sits a structure called the corbicula, a shallow concave surface fringed with curved hairs that together form a basket capable of holding a compacted pellet of pollen. Bees do not simply collect loose pollen and carry it home. They actively pack it. As a worker moves across a flower, it combs pollen from its body hairs. Using specialized combs on the middle legs, transfers the collected material to the hind legs, compresses it with a press-like structure, and adds a small amount of nectar or saliva to bind it. Together, the resulting pellet stays in place during flight, a tightly engineered cargo pod clipped to the body. A fully loaded worker returns to the hive with a visible orange, yellow, or occasionally dark red or white mass on each hind leg, the color determined by which flowers it visited. If you have ever watched a bee land on a flower and noticed what appears to be tiny colored saddlebags on its back legs, you were watching this entire collection and packing system in its final stage. The stinger of a worker honeybee is a modified egg-laying structure, an ovipositor repurposed over evolutionary time into a defensive weapon. It is not a simple spike. Under magnification, it reveals itself as a complex assembly of interlocking shafts, with backward-facing barbs along the shaft's length. When a worker bee stings a vertebrate, an animal with elastic, clenching skin those barbs catch, the bee pulling away cannot retrieve the stinger. It tears free from the bee's abdomen along with the attached venom sac and a cluster of nerve tissue which continues to pump venom and release alarm pheromone for several seconds after separation. The bee dies. This is important context. A worker bee does not sting lightly. The act is fatal to her. She performs it only as a last defense, and even then the instinct to sting is not as trigger-ready as popular understanding suggests. Drones, male bees have no stinger at all. Queens have one smooth and unbarbed, but rarely deploy it, except against rival queens. The famous sting belongs to the workers alone, and it costs them everything. A bee has 5 eyes. This is not a figure of speech or a loose approximation. The 2 large compound eyes on the sides of the head are the most visible, but positioned on the top of the head in a small triangle sit 3 additional eyes, called ocelli. These simple eyes do not form detailed images. They are photoreceptors tuned to detect light intensity and polarization, essentially. The bee's horizon level and sky reading instruments. They detect the polarization pattern of skylight, which changes predictably depending on where the sun is, even when the sun itself is hidden behind clouds. Bees use this information to maintain accurate bearings during navigation. Scientists still debate the full extent of what the ocelli contribute to flight stability and orientation, but experiments in which the ocelli are obscured show measurable impairment in the bee's ability to navigate under overcast conditions. 5 eyes, 3 functions, 1 seamless sensory system. The bee processes more dimensions of light information simultaneously than any vertebrate eye is capable of receiving. A bee's antennae are not decorative. They are among the most information-dense sensory organs in the animal kingdom relative to the size of the creature carrying them. Each antenna is lined with thousands of sensory receptors distributed across its length, some tuned to airborne chemicals, others to direct contact and surface texture, others still to airflow and vibration. The olfactory receptors alone number in the thousands, giving bees a sense of smell sensitive enough to detect floral compounds at concentrations that would register as essentially zero on most laboratory instruments. They can identify the species of a flower, assess whether it has already been visited and its nectar depleted, and recognize the chemical signature of nestmates versus intruders, all through antennal contact lasting fractions of a second. The antennae also detect the substrate vibrations that travel through comb inside the hive, a channel of communication that operates entirely below human hearing. When a bee cleans its antennae, a behavior performed with striking regularity, it is performing maintenance on instruments far more sophisticated than their delicate appearance suggests. A honeybee's tongue is not a tongue in the way mammals use the word. It is a proboscis, an extended, flexible tube-like structure that the bee deploys from a folded resting position into a working length of roughly 6 to 7 millimeters. It operates through a combination of capillary action and active lapping. The tip is fringed with fine hairs that sweep liquid upward, while the surrounding structures form a sealed channel through which the gathered nectar travels toward the bee's honey stomach. The system works something like a self-priming pump combined with a mop. A bee visiting a flower does not simply dip its tongue into the nectar pool and sip. It laps rapidly up to 5 times per second its proboscis in continuous oscillating motion. Nectar chemistry, viscosity and temperature all affect how efficiently the proboscis works, and bees are known to preferentially visit flowers whose nectar consistency matches the operating range of their mouthparts most closely. The tool selects the task, and the task has been refining the tool for millions of years. Bees do not have blood, not in the sense that vertebrates do. Their circulatory fluid is called hemolymph, a pale yellowish liquid that moves through an open circulatory system, rather than a closed network of veins and arteries. There is no oxygen delivery function in hemolymph. Oxygen reaches the bee's tissues through a separate system, entirely a branching network of air tubes called tracheae that carry air directly to the cells without any fluid intermediary. What hemolymph does carry is nutrients, hormones, waste products, and immune cells. It is pumped by a simple dorsal vessel, a tube running along the back of the bee that functions as a rudimentary heart, and it bathes the organs rather than circulating through enclosed channels. This open system is efficient enough for an insect body but would be catastrophically inadequate for anything larger. The reason insects cannot grow to the sizes imagined in old science fiction films is in part this very architecture. Beyond a certain body size, an open tracheal system cannot deliver oxygen fast enough to sustain life. Worker honeybees produce beeswax from their own bodies. On the underside of the abdomen sit 4 pairs of wax glands, specialized patches of tissue that in young workers between 10 and 16 days old convert consumed honey into tiny wax flakes secreted through pores in the cuticle. The bee then removes these flakes with its legs, chews them with its mandibles to soften and work the wax. and applies it to the comb under construction. The wax begins as a brittle, near-white material and is transformed through chewing into the warm, workable substance used to build honeycomb cells. Building comb is energetically expensive. Bees consume roughly 6 to 8 pounds of honey to produce a single pound of wax, a ratio that explains why established colonies reuse and repair comb rather than rebuilding it from scratch whenever possible. The hexagonal geometry of the finished cells has attracted the attention of mathematicians for centuries, because it achieves the maximum enclosed volume for the minimum material used, a solution that human engineers did not formally prove optimal until 1999. A queen honeybee and a worker honeybee begin as genetically identical eggs. What separates them is diet. A larva-fed royal jelly, exclusively throughout its larval development, becomes a queen. One switched to a mixture of pollen and honey after the first few days becomes a worker. The anatomical differences that result from this dietary divergence are substantial. A queen is noticeably longer than a worker. Her abdomen is extended to accommodate a pair of fully developed ovaries capable of producing up to 2,000 eggs per day. Her eggs lack the pollen baskets of workers. Her stinger is curved and smooth rather than barbed, allowing her to sting repeatedly without injury to herself. Her mandibles are shaped for fighting rival queens rather than manipulating wax. She has no wax glands. She will never forage, never build comb, never guard the entrance. From the moment she emerges from her cell, her entire physiological existence is organized around a single function: reproduction. Scientists still debate precisely how the royal jelly triggers such a comprehensive developmental divergence from an identical starting genome. A drone, a male honeybee, is built for one purpose, and his anatomy states this with unusual directness. His eyes are the most immediately obvious feature. They are so large they meet at the top of his head, wrapping nearly all the way around in a nearly seamless visual dome. This extreme eye development gives him the wide field, high-resolution vision needed to detect and pursue a virgin queen during a mating flight, which takes place high in the air and at considerable speed. He has no stinger. He carries no pollen baskets. He has no wax glands, no Nasonov gland, no foraging apparatus of any kind. He cannot feed himself from flowers inside the hive. He is fed by workers. He is larger than a worker, but moves with less precision, and the buzz his wings produce is noticeably lower in pitch due to his greater mass. His entire biological purpose is concentrated into a single mating event that, if successful, kills him. If he never mates, he lives through summer on the colony's resources and is evicted without ceremony and without exception when autumn arrives. A honeybee colony maintains the temperature of its brood nest, the area where eggs and larvae develop, within a range of roughly 34 to 36 degrees Celsius year-round, regardless of external conditions. This is not passive insulation. It is active collective climate control. In heat, bees fan their wings in coordinated airflow patterns to move hot air out of the hive. And some workers collect water to spread across the comb using evaporative cooling. In cold, bees cluster tightly together and vibrate their thoracic flight muscles, the same muscles that power flight-generating metabolic heat without moving. The cluster contracts as temperatures drop and expands as they rise, adjusting its surface to volume ratio to regulate heat loss. Individual bees on the cold outer surface of the cluster rotate inward to warm up, then move back to the surface, a slow, continuous circulation that prevents any individual from being exposed to lethal cold for too long. The precision of this collective thermostat, maintained without any central coordinator, has been a significant subject of study in the field of self-organizing systems for decades. Near the tip of a worker bee's abdomen sits a small structure called the Nasonov gland. When a bee exposes this gland, fanning her wings while bending her abdomen to present the gland opening to the air, she releases a blend of 7 chemical compounds that together constitute a homing and assembly signal. The scent is often described as having a faintly lemongrass quality, though bees are considerably more sensitive to it than any human nose would be. Scouts returning to a swarm cluster use the Nasonov signal to help other bees locate the cluster's position. Workers at a new hive entrance found Nasonov scent to guide returning foragers home. When a swarm finally commits to a new nest site, bees at the entrance fan this signal outward to draw the entire swarm inside. It is a landmark, a welcome signal, and a settling call, a single gland serving multiple social functions depending entirely on context. The bee does not decide which message it is sending. The context of the behavior carries the meaning. The family Apidae, which contains the honeybees most people picture when they hear the word bee, represents a narrow slice of a much larger world. There are approximately 20,000 known species of bee on Earth, ranging in size from the tiny Perdita minima, a native bee of the American Southwest, small enough to rest comfortably on a pencil eraser, to Wallace's giant bee of Indonesia, which carries a wingspan wider than an adult human thumb, is long, and was believed extinct for over a century before being photographed alive in 2019. Between these extremes sits extraordinary variety: bees that nest in snail shells, bees that are entirely black, bees that carry their pollen internally rather than in external baskets, bees that live solitary lives without any colony at all. Most bee species are solitary. The complex democratic society that makes honeybees remarkable is not the default state of bee life. It is an exceptional evolutionary achievement arrived at independently only a handful of times across the entire history of the order. The hive is not the rule, it is the exception that became the icon. A bee does not have bones. Its structural support comes from the outside, a hard external shell called the exoskeleton, made primarily from a material called chitin, layered with proteins, and in places waxes and pigments. This cuticle serves simultaneously as skeleton, armor, and skin. It supports the muscles, protects the organs, prevents water loss, and carries the sensory hairs and pores through which the bee reads its environment. Different regions of the cuticle are engineered to different specifications. The joints are thin and flexible, allowing movement. The compound eyes are covered by a transparent cuticular lens. The wing surface is an extraordinarily thin membrane, nearly weightless and yet mechanically resilient enough to survive thousands of flight cycles. The cuticle also carries species and colony-specific chemical signatures, hydrocarbon compounds that function as a chemical passport, readable by other bees through antennal contact, in the fraction of a second it takes to brush against a nestmate at the hive entrance. An intruder bee may look identical. It does not smell identical, and that difference is noticed immediately. A honeybee's color vision is shifted relative to human vision in a way that feels once understood, like standing in the same room, but looking through entirely different windows. Bees see well into the ultraviolet end of the spectrum, wavelengths completely invisible to the human eye, and are essentially blind to red, which appears to them as dark and indistinct as black. Their three color receptors are tuned to ultraviolet blue and green, compared to the human arrangement of blue, green, and red. This means a red poppy, vivid and saturated to human eyes, appears dull and near invisible to a bee approaching it from a distance. The poppy compensates by reflecting strongly in ultraviolet, a signal the bee reads clearly even when the human observer sees nothing remarkable. Many flowers have evolved elaborate ultraviolet patterns, nectar guides, target rings, directional streaks that exist in a visual dimension humans never see without specialized cameras. The meadow a bee flies over is not the meadow you walk through. Same plants, same light, entirely different visual world. A honeybee brain contains roughly 1 million neurons, a number that sounds large until you consider that the human brain contains approximately 86 billion. The bee's entire central nervous system, including both brain lobes and the nerve cord running through the thorax and abdomen, weighs less than a milligram. It is smaller than a sesame seed, and yet within this structure, bees navigate by sun, compass, memorize and communicate spatial information, recognize individual faces, learn abstract rules, demonstrate something resembling emotional states, and collectively make decisions that outperform many human groups. In accuracy and efficiency, the ratio of cognitive output to neural hardware in a bee is by any reasonable measure extraordinary. Neuroscientists studying insect brains have repeatedly revised their expectations downward in terms of minimum complexity required for a given behavior, and then revised them downward again. The bee brain is not a simple reflex machine. It is a compressed, highly efficient cognitive architecture that does more with less than almost any other known biological system. What the brain lacks in size, the colony compensates for in numbers, and together they produce something neither could achieve alone. When a scout bee returns to the hive after finding a good source of food or a promising nest site, she does something that took scientists decades to decode. She dances. The waggle dance is a figure-8 movement performed on the vertical face of the comb, and it contains precise symbolic information about a location that may be miles away. The angle of the straight central run of the figure-8, relative to vertical, corresponds directly to the angle of the food source, relative to the sun's current position. The duration of that central run encodes distance. Longer runs indicate greater distances. The vigor of the waggle, the number of circuits performed, communicates the scout's assessment of quality. A bee, watching the dance in the darkness of the hive, pressed close to the dancer, reading the vibrations through the comb beneath her feet, extracts all 3 pieces of information and can fly directly to the source without ever having seen it. This is symbolic referential communication about things displaced in space and time from the moment of communication. In the animal kingdom, only humans do this more elaborately. Scientists still debate whether this constitutes true language in the linguistic sense or something adjacent to it, remarkable but categorically different. Bees do not arrive in the world knowing which flowers hold nectar. They learn. Laboratory experiments have demonstrated that honeybees can be trained to associate abstract visual symbols, shapes, patterns, colors with food rewards, and that they retain these associations across multiple days and changing conditions. More striking still, bees can learn rules rather than just individual associations. When trained on a concept, such as choose the object that matches the sample bees, apply this rule to entirely novel objects they have never encountered, suggesting a capacity for generalization that goes well beyond simple conditioning. They can also learn the reverse, choose the object that does not match. These are the kinds of abstract cognitive tasks typically used to assess higher-order learning in vertebrates. The bee performs them reliably. What remains genuinely uncertain is whether the bee experiences anything resembling understanding, or whether its neural architecture achieves the same behavioral output through a fundamentally different internal process. The behavior is there. What produces it at the level of experience is a question that may never be fully answerable. Honeybees can count within limits. Studies in which bees were trained to choose a stimulus containing a specific number of elements, selecting 3 dots over 2 or 4 over 3, have found reliable discrimination of quantities up to 4. Beyond 4, accuracy drops significantly, which aligns with a pattern seen in many other animals and even in pre-counting human cultures. Small quantities are perceived directly. Larger ones require counting or estimation strategies that some animals simply do not have. Think of it like the difference between glancing at 3 keys on a table and knowing immediately how many there are, versus needing to actually count a pile of 17. Bees appear to inhabit the glancing end of that spectrum with reasonable precision. What makes the finding notable is not the upper limit of the ability, but the fact that the ability exists at all in an animal with a brain of this size. The neural resources required to distinguish quantity to treat threeness as a category separate from the specific objects being counted were not expected at this scale. They are there nonetheless. In 2018, a team of researchers published a finding that generated significant attention in cognitive science. Honeybees appear to understand the concept of zero as a quantity lower than 1. In experiments where bees were trained to select the stimulus with the smaller number of elements and then presented with a blank stimulus alongside numbered ones, bees chose the blank, the nothing more often than chance would predict, and did so more consistently when the comparison was against larger numbers. This behavioral pattern mirrors what has been observed in primates and young children learning numerical concepts, and suggests that representing nothing as a position on a numerical scale, rather than simply the absence of a thing, may not require the kind of language-dependent abstract thought humans typically associate with the idea. Scientists debate whether this constitutes a true conceptual understanding of zero, or a sophisticated but non-conceptual perceptual response. The distinction matters enormously in cognitive science. The bees, for their part, continue choosing the empty card, indifferent to the debate they have started. Honeybees show a measurable preference for turning in one direction over the other when navigating unfamiliar environments. Studies placing bees in maze-like structures, where they must choose between left and right at repeated decision points, have found a statistically significant tendency to favor the right turn, a bias that holds across individual bees and across different experimental setups. This kind of directional bias, sometimes called lateralization, was long considered a hallmark of vertebrate nervous systems associated with the division of the brain into functionally specialized hemispheres. Finding it in insects required a substantial revision of assumptions about what neural architecture is necessary to produce it. Lateralization in bees appears to extend beyond simple turning preferences. There is evidence of asymmetric antennal use in certain learning tasks, with the right antenna showing advantages in some contexts and the left in others. You might find it quietly remarkable that an animal with a brain smaller than a grain of rice has a dominant side, not so different in principle, from the hand you instinctively reach out with first. Honeybees do not use tools in the way a crow bends a wire or a chimpanzee strips a twig, but they employ a substance called propolis in ways that bear close comparison to strategic material use. Propolis is a resinous compound bees collect from tree buds and bark wounds carry back to the hive, mixed with wax and saliva, and apply throughout the colony's interior. Its antimicrobial and antifungal properties are well documented. The hive's propolis lining measurably reduces the bacterial load inside, functioning as a colony-wide immune defense built into the architecture itself. More striking is what bees do when a predator, too large to remove a mouse, for instance, dies inside the hive. Unable to extract it, bees encase the carcass entirely in propolis, effectively mummifying it and preventing decomposition from fouling the colony's air and food supply. This is not a reflexive behavior triggered by a simple stimulus. It is a response to a novel problem using an available material in a context different from its primary application. Whether that constitutes tool use, in a strict definitional sense, scientists still debate, but the pragmatic intelligence it represents is difficult to dismiss. A foraging honeybee does not treat every flower visit as a new event unconnected to previous experience. She remembers. Studies tracking individual foragers have shown that bees recall the location, species, reward quality, and even the time of day at which particular flowers were most productive, and they use this information to plan subsequent foraging trips. A patch of clover that yields rich nectar at mid-morning may be revisited at the same hour the following day, while being largely ignored at other times. This time-linked memory, knowing not just where a resource is, but when it is worth visiting, requires the simultaneous storage and retrieval of spatial, sensory, and temporal information bound together into a single retrievable episode. It is the kind of memory structure cognitive scientists call episodic-like memory, and it was for a long time considered a capacity unique to humans and a small number of other vertebrates. Bees were not on that list until relatively recently. The revision came, as many revisions in animal cognition do, from researchers willing to ask whether the behavior was present before, assuming it could not be. Bees maintain an internal clock of striking precision. In the early 20th century, the naturalist Auguste Forel noticed that bees began arriving at his breakfast table at the same time each morning, even on days when no food had yet been set out, drawn not by the smell of jam, but by the time of day itself. Subsequent research confirmed what Forel suspected. Bees possess a circadian rhythm accurate enough to be used as a foraging schedule. They visit flowers at the hours those flowers are most productive, and largely ignore them otherwise, even if the flowers are continuously available. This temporal precision is governed by the same kind of molecular clock mechanism found in most animals, including humans. But bees integrate it with spatial memory and reward history in a way that produces coordinated, timed foraging behavior across an entire colony. Individual foragers from the same hive may be assigned through dance communication to different flower patches, with different peak hours, each bee operating on its own internally scheduled shift, without any external timekeeper issuing instructions. The temperature regulation described earlier, in the context of bee physiology, is also, at a deeper level, a story about collective decision-making. No individual bee monitors the entire hive temperature. No single bee decides when fanning should begin, or when the water-carrying brigade should be deployed. Instead, each bee responds to local temperature information in its immediate vicinity, using simple behavioral rules. If it is too warm, fan. If it is too cool, cluster and vibrate. The colony-level outcome, a stable brood temperature maintained within 1 degree Celsius across varying external conditions, emerges from thousands of these local individual responses without any coordination from above. This is the same architectural principle that underlies many of the bee colony's most impressive collective achievements: local rules, local information, and a global outcome that no individual planned or perceives. Researchers studying self-organizing systems, from traffic flow to financial markets to immune responses, have used the bee thermostat as a model for understanding how complex, stable behaviors emerge from simple interacting components. The hive is not managed, it manages itself. Bees can learn to avoid food sources associated with harm, and this learning persists. Laboratory studies have shown that bees exposed to sublethal doses of certain pesticides during foraging subsequently show altered behavior, reduced return visits to the contaminated source, modified dance communication about it, and in some cases, apparent aversion even when the source is later presented without the chemical. This kind of aversive learning, forming a negative association with a stimulus after a harmful experience, is well established in vertebrates and has been demonstrated in bees with increasing rigor over the past 2 decades. What makes the finding practically significant is the implication that colonies in pesticide-affected landscapes may be modifying their foraging behavior in ways that are not immediately visible but measurably reduce their efficiency and nutritional intake. The bees are in a meaningful sense responding adaptively to a novel environmental threat. Whether they are responding adaptively enough, given the scale of pesticide exposure in modern agricultural landscapes, is a separate question, one that researchers and policymakers continue to work through without clear consensus. Within a honeybee colony preparing to swarm, a small subset of workers takes on a role that has no precise human equivalent. They become scouts. These are experienced foragers, older workers, who have already spent weeks navigating the landscape, and they volunteer, apparently spontaneously, to search for potential new nest sites rather than for food. A scout bee evaluating a candidate cavity inspects it with systematic thoroughness. She measures the volume by walking the interior. She assesses the entrance size, orientation, height from the ground, and the degree of existing weatherproofing. She checks for signs of previous occupancy, both by other bees and by predators. She may spend 30 minutes or more on a single inspection before returning to the swarm cluster to report. The dance she performs on her return is not a simple announcement. It is a calibrated advertisement whose intensity directly reflects her assessment of the site's quality, and she is prepared to dance for hours or even days to persuade others. She is not simply a messenger, she is an advocate. When a scout bee evaluates a potential nest site, she is not applying a single criterion. Research by Thomas Seeley and colleagues identified at least 5 variables that scouts assess and weight when forming their quality judgment: cavity, volume entrance, size entrance, height from the ground, the degree to which the cavity is already weatherproofed, and its compass orientation. The optimal nest site from a honeybee colony's perspective has a specific volume range large enough to store sufficient honey for winter, but not so large it cannot be defended, and heated a small south-facing entrance positioned low in the cavity wall, significant pre-existing insulation and reasonable height above the ground to reduce predator access. Scouts synthesize these variables into a single quality assessment that is then communicated through the intensity and duration of their waggle dance. The entire evaluation process, conducted by an insect with a sub-milligram brain, produces a multivariable weighted score, the same kind of decision architecture that human engineers implement deliberately in formal evaluation frameworks. The bees arrived at it without a framework document. Bee swarm democracy does not work by majority vote. It works by quorum. As scouts return from their inspections and begin dancing for their preferred sites, other scouts are recruited to visit and independently assess the same locations. Over time, the best sites attract the most dancers, and the dancing for inferior sites gradually fades as scouts are converted to better options or simply stop dancing. But the swarm does not move the moment one site has more dancers than another. It waits. The swarm moves only when the number of scouts simultaneously present at a single site reaches a threshold, estimated at roughly 15 bees, that signals sufficient consensus has been achieved. This quorum requirement serves a critical function. It prevents the swarm from committing to a site based on premature enthusiasm. A site that initially attracts many dancers but cannot sustain a quorum is filtered out. The threshold is not fixed by any individual, it emerges from the dynamics of the recruitment process itself. Speed and accuracy are both served by this mechanism, which turns out to be more resistant to error than either pure voting or pure consensus systems. The bee swarm's decision-making process includes not just positive advocacy dancing for a preferred site, but active inhibition of competing options. Scouts who have visited multiple sites and formed a preference for one will, upon encountering scouts dancing for a different location, deliver a stop signal, a brief, vibrational headbutt that causes the dancing bee to pause or cease her dance entirely. This stop signal is not aggressive in the physical sense. It is informational, a correction injected into the deliberation process by a bee whose own assessment contradicts the one being advertised. The effect is to reduce the rate at which inferior options accumulate dancers, accelerating convergence toward the best available site, without requiring any scout to directly compare all options simultaneously. The mechanism is, in structural terms, very similar to inhibitory signaling in neural networks, where neurons suppress competing signals to sharpen the clarity of a final output. Whether this parallel reflects deep structural similarity between hive decision-making and brain function, or simply the same mathematical logic arriving at the same solution in different substrates, is a question researchers find genuinely interesting and not yet fully resolved. bee swarm democracy is not perfect. Occasionally, scouts make poor assessments. A site that appears adequate on first inspection may have flaws that only become apparent under closer scrutiny. Occasionally, a mediocre site gains early momentum through chance and threatens to pull the swarm toward a suboptimal choice. The system has a correction mechanism, because scouts continue to independently visit and reassess sites throughout the deliberation period. Rather than simply accepting the word of earlier assessors, poor sites tend to lose their dancers over time as new visitors form. Lower quality assessments and reduce their dance investment. The process is self-correcting, not through any individual's authority to overrule others, but through the cumulative weight of repeated independent verification. Seeley has described this as one of the most elegant features of the system. It achieves accuracy not by being right the first time, but by being structured so that errors are naturally depressed and good information naturally amplified. Human deliberative bodies have on occasion attempted to engineer similar correction mechanisms. Bees evolved one without trying. A foraging honeybee routinely travels up to 5 kilometers from her hive in search of food. A radius that, scaled to her body size, is roughly equivalent to a human walking from one end of a mid-sized city to the other and back multiple times per day without a map, a phone, or a street sign. She navigates this territory using a memorized map of visual landmarks, trees, hedgerows, buildings, changes in terrain, the shape of the horizon at different compass bearings. This map is not innate. It is built through a series of orientation flights performed early in a young bee's foraging career, during which she flies in expanding arcs around the hive entrance, facing back toward it, committing visual reference points to memory. In relation to her home, studies in which landmarks are moved or removed show measurable disorientation and increase navigation errors, confirming that the landmarks are genuinely being used rather than serving as incidental background. The map a bee carries is spatial, relational, and updatable, a cognitive achievement that requires at minimum the ability to encode, store, and retrieve complex multi-element visual information in sequence. Bees navigate using the sun as a directional reference, but the sun moves. Across a foraging day, it arcs from east to west, shifting its azimuth continuously, and a bee that simply pointed toward the sun would be traveling in a perpetually changing direction. Bees compensate for this movement through an internal time-corrected sun compass. They track the sun's rate of movement across the sky and adjust their directional calculations accordingly, even when foraging trips extend over hours. More remarkably, bees can extrapolate the sun's position when it is obscured by clouds, using the polarization pattern of skylight, which changes predictably with the sun's position to infer where the sun is, even when they cannot see it directly. They can also, to a degree, predict where the sun will be after a period spent inside the dark hive, updating their navigational bearing before they emerge. The waggle dance encodes all of this compensation. The angle danced inside the hive in the dark on a vertical surface already accounts for the sun's movement during the time the scout spent traveling. There is accumulating evidence that honeybees can detect Earth's magnetic field. Iron-containing compounds, specifically magnetite crystals, have been identified in the abdomens of honeybees in locations and orientations consistent with magnetoreceptive function. Behavioral experiments have shown that bees, under certain conditions, orient their comb construction in alignment with the geomagnetic field, and that disrupting the local magnetic environment with artificial magnets produces measurable changes in hive behavior. Bees exposed to altered magnetic fields during the waggle dance show reduced accuracy in the direction information they communicate. The mechanism by which this magnetic information is processed and integrated with the bee's other navigational systems remains incompletely understood, and the field is active with competing hypotheses about which cells or structures are actually doing the sensing. What seems clear is that the bee's navigational toolkit is larger than the sun compass and landmark memory alone. It may include a geomagnetic sense that provides a stable directional reference, independent of light conditions, weather, or time of day. In 2011, a research team at Newcastle University published a study that provoked considerable discussion in behavioral science. Honeybees, subjected to simulated predator attacks, shaken vigorously in a manner designed to mimic being seized, subsequently showed a measurable shift in how they responded to ambiguous stimuli. Bees trained to associate one odor with reward and another with punishment were, after the stressful shaking, more likely to interpret a novel intermediate odor as predictive of punishment rather than reward. This pessimistic cognitive bias, interpreting ambiguous information negatively following stress, is one of the behavioral markers used to infer negative emotional states in vertebrates. Its presence in bees does not prove that bees experience suffering in any conscious sense. The question of insect subjective experience remains genuinely open, and researchers are careful about overreaching. But it does suggest that whatever internal states bees have are not simply neutral processing outputs. They appear to be valenced, influencing subsequent behavior in ways that parallel the function of emotion in other animals. In 2022, researchers studying bumblebees, close relatives of honeybees, published footage and data showing young bees repeatedly rolling small wooden balls with no apparent functional purpose, no food reward, no social context, no obvious survival benefit. The behavior was spontaneous, was performed more often by younger bees, and showed individual variation in enthusiasm. Some bees returned to roll the same balls repeatedly over multiple days. The researchers were cautious about calling it play. In the full sense the word carries for mammals, but the behavioral criteria typically used to define play, intrinsically motivated, repeated, performed in a relaxed state, non-functional, in immediate terms were met. Similar, spontaneous, apparently non-functional behavior has been observed in honeybees under certain conditions. Whether this constitutes something like enjoyment or a developmental behavior that serves a function not yet identified remains uncertain. What it clearly represents is behavior that does not fit the old model of insects as purely reactive automatons executing fixed programs with no internal variation. The balls roll. The bees return to them. Sleep in bees is not simply the absence of activity. Sleeping bees and bees do sleep in postures and durations that are measurable and consistent, sometimes exhibit brief involuntary movements during their rest antenna, twitches, leg tremors, small whole-body jerks that occur without any apparent external trigger. In 2024, researchers studying sleeping bees with high-resolution imaging found that these movements were associated with rapid changes in antennal position that bore a structural resemblance to the rapid eye movements that characterize REM sleep in mammals, the sleep stage most associated with dreaming. In humans, the researchers also observed what appeared to be color changes in the bees' compound eyes during these episodes, suggesting altered neural activity. Whether bees dream in any meaningful sense is a question that cannot currently be answered, because answering it would require access to subjective experience that no external measurement can provide. What can be said is that bee sleep is not uniform, that it contains stages, and that something is happening during those twitching intervals that is not simple quiescence. Young honeybees do not develop foraging skills in isolation. Before making their first solo foraging trips, they spend time near the hive entrance, observing returning foragers, and within the hive they attend waggle dances performed by experienced scouts. Studies have shown that naive bees exposed to dances for a specific flower type preferentially visit that flower type on their early independent foraging trips, an effect that persists even when the flower is not the most rewarding option available in the environment. This transmission of foraging preference through observation and dance attendance constitutes a form of social learning. Information acquired by one individual encoded in behavior and transmitted to others, who then act on it without direct experience of the original source. The colony's collective knowledge about the local landscape, which flowers are blooming where, and how rewarding they are, is maintained partly through this social transmission system, constantly updated as experienced foragers return and dance, and as younger bees absorb and act on that information. Bees do not simply abandon a foraging route when an obstacle appears. Studies presenting trained foragers with novel barriers, screens, mazes, physical blockages placed along established flight paths have found that bees actively explore alternative routes, eventually finding detours that allow them to reach their target. This rerouting is not random. Bees appear to use their spatial memory of the landscape to evaluate alternative paths, preferring detours that minimize total distance traveled and that maintain directional alignment with the target when possible. The speed and efficiency of this rerouting improves with experienced bees that have been blocked and rerouted before, solve subsequent obstacles faster than bees encountering their first barrier. What this demonstrates is not just that bees can navigate around obstacles, many animals do this through simple trial and error, but that they appear to draw on a stored spatial representation of the environment to plan rather than simply react. The distinction between planning and reacting is one of the more contested lines in animal cognition, and bees sit somewhat uncomfortably for old frameworks on the planning side of several experiments. A honeybee colony accumulates knowledge that no individual bee possesses in full. An experienced forager knows her own flight routes and flower patches. A scout who evaluated nest sites last season carries those assessments. A nurse bee knows the developmental state of the brood she tends. A guard bee knows the current chemical signature of her colony. None of these individuals holds more than a fragment of the colony's total information. But the colony as an entity holds all of it simultaneously distributed across thousands of individuals, accessible through dance communication, pheromonal signaling, and direct physical interaction. When individual bees die, and they die constantly, workers living only weeks during summer, the information they carried is lost, but the colony's overall knowledge base remains largely intact, because it was never concentrated in any one place. This distributed architecture makes the colony's memory extraordinarily robust to individual failure. It is structurally closer to how the internet stores data redundantly across many nodes, with no single point of catastrophic failure, than to how any individual brain stores experience. Thomas Seeley, the Cornell biologist who has spent decades studying bee swarm intelligence, has argued that the honeybee colony is best understood not as a collection of individual insects, but as a single cognitive entity, a superorganism, whose collective behavior constitutes something analogous to thought. The individual bee, in this framing, is less like a person and more like a neuron, a simple unit whose individual behavior is limited, but whose interactions with thousands of similar units produce outcomes of genuine complexity and adaptability. The swarm's decision-making process, independent information, gathering, parallel evaluation, inhibitory signaling, quorum sensing, error correction, mirrors in structural terms. The process by which a primate brain integrates competing sensory inputs and arrives at a decision. Whether this parallel is deep or superficial is a question that touches on some of the most fundamental, unsettled issues in cognitive science. What decision-making actually requires at a physical level, and whether the substrate neurons, or bees, changes the nature of what is happening. The hive thinks, in some sense. What kind of sense that is remains genuinely open. The word colony undersells what a honeybee hive actually is. Biologists use a different term, superorganism. A superorganism is a collection of individuals so thoroughly integrated in their physiology, behavior, and communication that the collective functions as a single living entity in ways that the individuals alone cannot. A honeybee removed from her colony does not simply struggle, she deteriorates. Her immune function declines. Her behavioral repertoire narrows. Her lifespan shortens dramatically. She is not, in any complete biological sense, a standalone organism. She is a component, and components do not function well in isolation from the system they belong to. The colony breathes as one, regulating its own internal atmosphere through coordinated fanning. It maintains temperature as one. It makes decisions as one. It reproduces as one. Through swarming, individual workers are mortal and replaceable. The colony can persist for decades, replacing every component while maintaining its identity, its location, and its accumulated knowledge. This is not a metaphor for cooperation. It is a literal description of a biological organization level that sits between the individual organism and the species. The queen honeybee governs her colony not through command, but through chemistry. She continuously produces a blend of chemical compounds, collectively called queen mandibular pheromone, that workers collect from her body during grooming and spread through the colony via trophallaxis, the direct mouth-to-mouth transfer of food and fluid. This pheromone blend carries a remarkable amount of information. It suppresses the development of worker ovaries, preventing workers from reproducing. It inhibits the rearing of new queens when the colony is stable. It influences worker behavior, keeping the colony calm and cohesive. It attracts drones during mating flights. It signals the queen's presence and health to every bee in the colony simultaneously. A colony that loses its queen detects her absence through the fading of this chemical signal within hours and responds by beginning emergency queen rearing within a day. The pheromone is not a simple on-off signal. It is a continuously updated broadcast of the queen's physiological state, and the colony reads it with the same continuous attention that a healthy body pays to its own hormonal environment. All female honeybees begin as identical eggs. The developmental path that follows, whether a larva becomes a queen capable of reproduction or a worker who will spend her life foraging, building, guarding, or nursing, is determined not by genetics but by nutrition, specifically by what she is fed during the first days of larval life. Worker larvae receive royal jelly initially, then transition to a mixture of pollen and honey called worker jelly. Queen larvae receive royal jelly continuously throughout larval development. Within this apparently simple dietary difference lies an extraordinary epigenetic mechanism. The compounds in royal jelly alter the expression of genes across hundreds of developmental pathways, producing an animal that is anatomically, physiologically, and behaviorally distinct from her genetically identical sisters. Among workers, further differentiation occurs through age-based task progression. A young worker begins as a nurse, transitions through wax production, guard duty, and foraging over the course of her adult life with her internal physiology, shifting at each stage to match her current role. The same individual is, in a meaningful sense, several different kinds of worker across the span of a single life. Royal jelly is produced by young worker bees from glands in their heads, specifically the hypopharyngeal glands, and it is among the more chemically complex substances produced by any insect. It is a white, slightly acidic substance with a sharp, tangy smell composed of water, proteins, sugars, fatty acids, and trace compounds whose full functional significance is still being mapped. The key active component appears to be a protein called royalactin, which was identified as a primary driver of queen development in a 2011 study, though subsequent research has complicated this picture, suggesting that the full developmental effect involves multiple interacting compounds, rather than a single trigger. Royal jelly's effects extend beyond the larva that consumes it. A queen continues to be fed royal jelly throughout her adult life and there is evidence that this ongoing consumption contributes to her dramatically extended lifespan. A worker bee lives 4 to 6 weeks in summer. A queen-fed royal jelly from larva to death may live 4 to 5 years. Same genome, different diet, a lifespan more than 30 times longer. For the first 10 days or so of her adult life, a worker honeybee's primary occupation is caregiving. Nurse bees tend the brood, the developing eggs, larvae and pupae in their wax cells, with a level of attentiveness that in aggregate amounts to thousands of individual inspections per day across the colony. Each larva is visited by nurse bees up to 1,300 times over the course of its development. Nurses assess the larva's condition, deliver the appropriate food in the appropriate quantity, adjust feeding based on developmental stage, and maintain the humidity and temperature of the brood area through their own body heat and behavioral adjustments. They also perform quality control. Larvae that are diseased, parasitized, or developmentally compromised are identified and removed from the comb before they can affect neighboring brood. A behavioral immune response called hygienic behavior that varies in effectiveness between colonies and has been the subject of selective breeding programs aimed at producing disease-resistant stock. The nurse bee does not decide to care. She is, for this stage of her life, constitutionally a caregiver. Her glands are active, her behavior is calibrated, her attention is fixed on the comb. Death is a constant presence inside a honeybee colony. Workers die at rates of hundreds per day during peak summer, and the hive's response to each death is swift, specific, and handled by a small subset of workers who appear to specialize in corpse removal. These undertaker bees, typically older workers, detect the chemical signature of death, oleic acid and related compounds, released by a dead bee's decomposing tissues, the same class of chemicals that serve as death signals in many social insects. Upon detecting these compounds, an undertaker removes the corpse from the hive within 30 minutes on average, carrying it a significant distance from the entrance before dropping it. The efficiency of this removal is not merely aesthetic. A decomposing body inside the hive represents a vector for pathogen spread, and rapid removal measurably reduces disease transmission. Undertaker bees are not a permanently designated caste. Individual workers transition into and out of undertaking behavior based on the colony's current needs and the chemical environment inside the hive. A bee that has been handling corpses can be induced to resume nursing behavior if the colony's nurse population drops. The roles are firm. The role holders are flexible. At the entrance of every established honeybee colony stand guard bees workers who have reached the age at which their venom glands are fully developed and who position themselves at the hive entrance to screen incoming bees. The screening is chemical. Every honeybee colony has a distinctive blend of cuticular hydrocarbons, waxy compounds on the surface of every bee's exoskeleton that functions as a colony-specific chemical passport. A guard bee inspects each returning forager through brief antennal contact, comparing the chemical signature she detects against the template she carries. Nestmates pass, strangers are challenged, and if they cannot present an acceptable chemical profile, they are physically repelled, or in cases of persistent intrusion, stung. The system is not infallible. Robber bees from other colonies sometimes breach it through behaviors that appear to mimic nestmate chemical profiles. But under normal conditions, it maintains the colony's boundary with remarkable reliability. Guards also respond to alarm pheromone released by stinging bees, recruiting additional defenders rapidly when the entrance is threatened. The transition from routine inspection to defensive mobilization takes seconds. The waggle dance is not performed to an empty room. When a scout returns to the swarm cluster and begins dancing, she is immediately surrounded by follower bees who press close, maintaining antennal contact with the dancer, tracking her movements through the vibrations she transmits into the pontic substrate beneath their feet. These followers are not passive observers. They are evaluating. A follower bee who finds a dance sufficiently compelling may herself fly out to inspect the advertised site, and if her independent assessment confirms the scout's quality rating, she returns and adds her own dance to the growing chorus of advertisement for that location. A follower who finds the site disappointing dances less vigorously or not at all. The dance floor of the swarm, the surface of the cluster where dozens of scouts may be simultaneously dancing for different sites, is thus a living dynamic market of competing information, where quality is not simply announced, continuously tested against independent verification. The audience is the error correction mechanism. No single dancer's assessment goes unexamined. The complete sequence of a honeybee swarm decision unfolds over 1 to 3 days and follows a progression of remarkable regularity. The swarm departs the parent hive and settles into a temporary cluster, typically on a nearby tree branch, within minutes. Scouts, drawn from the experienced foragers in the swarm, immediately begin departing to search for nest sites. They return and dance. Followers are recruited. Sites are visited, assessed and danced for, or abandoned. Stop signals suppress inferior options. The best site accumulates dancers faster than its competitors. When the quorum threshold is reached at the winning site, scouts at that location begin performing a piping signal, a vibration transmitted through the cluster that triggers the swarm's departure. The entire cluster lifts off and navigates to the new site, guided by a small number of fast-flying streaker bees who know the destination and fly through the swarm in the correct direction, providing directional information to the mass of bees who do not. The colony arrives. Workers begin building comb within hours. A new colony exists where none did before. Established through a process that produced no losers, extracted no confession, and required no authority. When a honeybee queen begins to fail, her pheromone output declining, her egg-laying rate dropping, her chemical signal to the workers growing faint, the colony does not wait for her to die before acting. Workers begin constructing queen cells, large peanut-shaped wax structures built onto the face of the comb that are provisioned with royal jelly and seeded with eggs or young larvae. The queen who will replace the failing one is raised in secret, so to speak, her development proceeding, while the current queen is still alive and laying. When the new queen emerges, one of two things happens. The old queen departs with a swarm before the new one emerges, or the new queen kills any rival queen still in their cells by stinging through the wax, and then, if the old queen is still present, a fight occurs that ends with one queen dead. Workers sometimes appear to facilitate this outcome by retaining rival queens in their cells until the strongest has emerged and established herself. The process is efficient, occasionally brutal, and entirely worker-managed. The workers do not mourn the old queen. They have already prepared her successor. When a honeybee colony loses its queen and fails to replace her in time, when no viable eggs or young larvae remain from which a new queen can be raised, the colony enters a slow, irreversible decline that begins with a biological rebellion. Without the queen's pheromone suppressing their reproductive development, the ovaries of worker bees begin to activate. Workers start laying eggs. This sounds on the surface like an adaptive response, the colony attempting to reproduce in the absence of its reproductive specialist. It is not. Worker bees are unmated. Their eggs, being unfertilized, can only develop into drones males, who will contribute nothing to the colony's survival and will consume its resources while the worker population dwindles with no replacements. A laying worker colony is a colony in the process of dying, and the process is difficult to reverse because the laying workers now produce pheromones that mimic imperfectly the queen's suppressive signal, making the introduction of a new queen extremely difficult, as workers conditioned by these ersatz pheromones are likely to reject and kill her. Beekeepers who manage colonies through this crisis describe it as one of the more sobering demonstrations of what the queen's chemistry actually holds together. Male honeybees' drones do not mate near the hive. They gather in specific aerial locations called drone congregation areas zones, in the landscape typically positioned between tree lines or at the edges of open terrain where drones from many different colonies assemble in dense buzzing clouds and wait. These congregation areas are not random. They are used generation after generation, returning to the same airspace year after year, even as the individual drones who use them are replaced entirely each season. How drones locate and return to the same areas remains incompletely understood. Magnetic cues, visual landmarks, and pheromonal gradients left by previous gatherings have all been proposed. A virgin queen who flies into a congregation area may mate with up to 15 or 20 drones during a single mating flight, storing sperm from multiple males in a specialized organ called the spermatheca. She will use this stored sperm to fertilize eggs for the rest of her life, potentially 5 years, never mating again. The congregation area is in effect the colony's genetic exchange point, a fixed location in the landscape where the hereditary futures of multiple colonies are shuffled together season after season invisibly. When a honeybee stings, she releases, along with the venom, a chemical signal from a gland near the stinger isoamyl acetate, the primary component of bee alarm pheromone. Its scent, to those sensitive enough to detect it, has been described as faintly banana-like, an incongruous sweetness attached to a signal that means something quite different to bees. In the vicinity of a threatened hive, this compound spreads through the air in seconds and triggers a rapid specific response in any worker who encounters it. Heightened alertness, increased aggression, and orientation toward the source of the signal. Additional bees are drawn to the location and primed to sting. The alarm pheromone is self-amplifying. Each subsequent sting releases more, which recruits more defenders, which produces more stings. This cascade is the mechanism behind the seemingly disproportionate response a honeybee colony can mount to a perceived threat. What begins as a single defensive sting becomes within moments a coordinated chemical military response. Beekeepers use smoke at the hive entrance, partly to mask this pheromone, breaking the alarm cascade before it builds. The smoke does not calm the bees. It interrupts their communication. Inside the hive, food moves between bees, not just through storage in cells, but through direct mouth-to-mouth transfer called trophallaxis. A returning forager carrying nectar in her honey stomach regurgitates it to a house bee waiting near the entrance, who passes it to another, who may pass it again before it reaches a storage cell. This chain of transfers is not merely logistical. Each exchange involves antennal contact, brief chemical inspection, and the transfer of not just food but dissolved pheromones, immune compounds, and microbiome components along with the liquid. Trophallaxis is simultaneously a feeding system, a communication channel, a disease resistance distribution network, and a social bonding behavior. Studies tracking individually marked bees have shown that food introduced at one point in a colony is distributed to a significant fraction of the colony's members within hours, meaning that every bee in the hive is, chemically speaking, in continuous contact with every other through this liquid network. The hive does not simply share food, it shares through food, a continuous chemical conversation that keeps every member updated on the colony's collective physiological state. Honeybees do not live in sterile environments, but they maintain their hives with a degree of microbial control that has attracted serious attention from medical researchers. Propolis, the resinous material collected from tree buds and bark wounds, is applied throughout the hive interior as a thin coating on walls, comb surfaces, and the interior of brood cells. Its antimicrobial properties are not incidental. Propolis contains dozens of biologically active compounds, including flavonoids, phenolic acids, and aromatic aldehydes, many of which show demonstrable activity against bacterial and fungal pathogens in laboratory conditions. Colonies with higher propolis deposition show measurably lower pathogen loads and stronger immune gene expression in individual workers, as though the architectural coating reduces the infectious burden enough that individual bees need to mount less vigorous immune responses. The composition of propolis varies by geography. Because it is derived from local plant resins, bees in temperate forests produce chemically different propolis from bees in tropical regions. The colony's medicine cabinet is stocked from its immediate landscape, and its contents change with the seasons as different trees and plants become available. When ambient temperature drops below roughly 14 degrees Celsius, a honeybee colony forms a winter cluster, a dense, roughly spherical mass of bees packed together on the comb. The queen at its warm center workers, arranged in layers from a warm inner core to a cooler, densely packed outer shell. The cluster is not static, it pulses. The outer bees vibrate their flight muscles continuously to generate heat, then rotate inward to warm up as their energy reserves deplete, while inner bees cycle outward to take their turn at the cold shell. The cluster moves slowly across the comb over winter, consuming the honey stored in cells immediately surrounding it, a slow, deliberate migration through its own food supply. A cluster that runs out of honey on one side of the comb and cannot move to where honey remains on the other side because the temperature difference between the two locations is too great to cross, can starve in proximity to full honey stores. Winter colony losses attributed to starvation often follow this pattern. The colony did not run out of food, it ran out of warmth sufficient to reach it. The waggle dance is not identical across all honeybee populations. Different subspecies of Apis mellifera, the western honeybee, show measurable variation in the relationship between dance duration and the distance communicated. Italian bees, Carniolan bees, and African bees each have their own calibration. A dance of a given duration encodes a different distance depending on which population is performing it. When bees from different subspecies are mixed in a single experimental colony and must read each other's dances, communication errors occur. A bee following an Italian dancer's instructions and expecting to find a food source at the distance that duration would indicate in her own dialect, arrives at the wrong location. These dialectal differences are genetically encoded rather than culturally learned, suggesting they evolved independently in geographically isolated populations over thousands of generations. The finding has implications for understanding how communication systems evolve. The dance is not a universal language, even within a single species. It is a family of related languages, each shaped by the particular landscape and foraging ecology of the population that developed it. The emergence of a new bee from its pupal cell is not always unassisted. Workers have been observed actively helping emerging bees break through the wax capping of their cells, chewing at the wax from the outside, creating openings that make emergence easier for bees who are struggling. This behavior is most commonly observed in colonies with high hygienic behavior scores, where workers are already predisposed to interact frequently with the broodcomb. Whether this constitutes genuine midwifery assistance calibrated to the emerging bee's need, or a more reflexive response to the vibrations and chemical signals produced by a bee in the process of emergence, is a distinction researchers have not yet fully resolved. What is documented is that assisted emergence does occur, that it is not universal, and that the bees who perform it show individual variation in the frequency with which they do so. The hive's care for its young does not end at the larval stage. It extends in at least some colonies, and under some conditions all the way through to the moment of birth. At any given moment inside the hive, the queen is surrounded by a group of 8 to 12 worker bees, called the retinue attendants, who groom her, feed her, and collect the pheromones from her body that will be distributed through the colony. Retinue bees face the queen, oriented toward her, touching her with their antennae and mouth parts, in a continuous cycle of chemical sampling and food transfer. Individual workers remain in the retinue for only a few minutes before being replaced by others, creating a constantly refreshed ring of attendants around the queen throughout her active hours. The function of the retinue extends beyond basic care. Each attendant, upon leaving the queen and moving through the hive, carries queen pheromone on her body and distributes it through trophallaxis and direct contact with other workers. The retinue is thus the distribution mechanism for the queen's chemical governance, a living broadcast network that carries her signal from its source to every corner of the colony. Without the retinue, the queen's pheromone would not reach the far ends of a large hive in concentrations sufficient to maintain its suppressive effects. For most of the active season, drones are fed, housed, and tolerated inside the hive, despite contributing no labor to its maintenance. As summer ends and nectar sources diminish, this arrangement terminates, and the termination is neither gradual nor gentle. Workers begin blocking drones from accessing honey stores. They physically drag drones toward the hive entrance. Drones who resist or attempt to return are refused entry. Within days, all drones in the colony are expelled, and those that cannot find food outside, which is all of them since drones are incapable of self-sufficient foraging, die of exposure and starvation. The eviction is consistent across colonies and precisely timed to the colony's assessment of approaching winter, triggered by declining day length, falling temperatures, and diminishing forage availability. It is one of the clearest examples of collective resource management in the animal kingdom, a unanimous decision executed without deliberation to shed a significant portion of the colony's population that provides no winter survival benefit. The drones do not fight the decision. They have no mechanism to. The workers have already made up their minds. A honeybee colony can leave its home in two fundamentally different ways, and the distinction between them reveals something about how the colony reads its own situation. Swarming is a reproductive act. The colony has grown strong enough that it can afford to split, sending the old queen and roughly half the workers to find a new home, while the remainder stays behind with a new queen. It is a sign of abundance. Absconding is different. When a colony absconds, the entire population leaves. Every bee, the queen, whatever honey, can be carried in their stomachs, abandoning the nest entirely. No population stays behind. No resources are left. Absconding is triggered by conditions that make the current location untenable: persistent predator pressure, severe parasite infestation, disease, chronic food shortage, or repeated disturbance. African honeybee subspecies abscond more readily than European ones, a difference that reflects the more intense predator pressure and more variable forage availability of their native landscape. The colony that absconds is not fleeing in panic. It is executing a collective decision that the current location has become a liability, and that survival requires starting over somewhere else entirely. The relationship between flowering plants and bees is not one of simple exploitation: flowers using bees as unwitting couriers, bees raiding flowers for their resources. It is a partnership refined across roughly 80 million years of mutual pressure, each party shaping the other's evolution in response to what the other offered or demanded. Flowers evolved colors, shapes, scents, and nectar compositions that specifically attract and reward bees. Bees evolved body forms, sensory systems, and behavioral repertoires specifically suited to accessing and processing what flowers offer. The fit between a bee's tongue length and the depth of the flowers it preferentially visits is in many cases so precise that the two appear designed for each other, which, in the only meaningful biological sense of that word, they were. This co-evolutionary relationship is not merely beautiful, it underpins the reproduction of the majority of flowering plant species on Earth, and through those plants, the food supply of most terrestrial ecosystems. The bee is not a visitor to the flowering world. It is in a deep sense one of the forces that built it. Asian honeybees, specifically Apis dorsata, the giant honeybee of South and Southeast Asia, have developed a collective defense behavior against predatory wasps that has no direct equivalent in Western honeybee species. When a wasp approaches the exposed comb of a dorsata colony, these bees nest in the open on cliff faces and tree branches. Without the enclosure of a cavity, workers on the comb surface respond by flipping their abdomens upward in a wave that propagates across the colony's surface like a ripple across water. Thousands of bees participate in milliseconds, the wave traveling faster than any individual bee could initiate and sustain alone. The effect to a predatory wasp is a disorienting, shimmering visual display that makes it extremely difficult to identify and target a single bee. The surface appears to be in continuous, unpredictable motion. Scientists studying this behavior have found that it is triggered by visual input at the colony's periphery and propagates through a simple neighbor response rule. Each bee flips when it sees its neighbor flip. No central coordination. A colony-level defense built from a single bee behavioral rule repeated 50,000 times simultaneously. Honeybees can learn to distinguish between photographs of individual human faces and retrieve specific faces from memory across multiple days. This finding, which emerged from a series of experiments in the early 2000s and has been replicated and extended since, was initially surprising because human face recognition is typically understood as a highly specialized cognitive capacity, requiring dedicated neural machinery. In humans, a specific region of the brain called the fusiform face area. Bees have no such structure. What they appear to do instead is apply a general configural processing strategy, assessing the spatial relationship between elements of a visual pattern that happens to work on faces as well as on flowers and landmarks. The bees are not recognizing faces the way humans recognize faces, but the behavioral output is functionally similar. They can tell individuals apart, remember which individual was associated with a reward, and make that discrimination reliably across changing conditions. Whether you find that reassuring or slightly unnerving may depend on how often you stand near beehives. The interior of a honeybee hive is not silent. It is filled with a continuous layered acoustic environment that bees both produce and read. Workers communicate through the comb via substrate-borne vibration signals, transmitted not through the air, but through the wax and wood of the hive structure itself. The waggle dance is accompanied by specific vibrational pulses that followers detect through their legs. The stop signal is a brief burst of vibration delivered through physical contact. Queen piping the sound a newly emerged queen makes travels through the comb and can be detected by the entire colony simultaneously. The tooting and quacking calls of queens still in their cells, answered by the emerged queen above, constitute a dialogue that workers monitor and respond to. Researchers recording the hive's acoustic environment have found that experienced beekeepers can distinguish by ear a queen-right colony from a queenless one. The collective sound shifts in frequency and character when the queen's presence is removed. The hive speaks in vibration as fluently as it speaks in chemistry, and the two languages woven together through every surface and every body in the colony carry the continuous conversation of 50,000 lives lived in extraordinary proximity to one another and to something very much like purpose. The oldest confirmed bee fossil, a specimen of Cretotrigona prisca preserved in amber from what is now New Jersey, dates to approximately 65 to 70 million years ago, placing it at the boundary of the Cretaceous and Paleogene periods. But molecular clock analyses, which estimate divergence times from genetic data, suggest the bee lineage is considerably older. possibly originating around 100 million years ago in the Cretaceous, when flowering plants were themselves diversifying rapidly across the planet. The amber specimens that survive from this period are extraordinary objects, insects, frozen in the exact posture of their final moment, sometimes with pollen grains still visible on their bodies, preserving in 3 dimensions the anatomy of creatures that lived when dinosaurs still walked on every continent. Holding one of these specimens, or looking at a photograph of one, which is the more realistic option for most people, is an encounter with the deep past that no written description quite prepares you for. The bee in the amber was already a bee, already social, already, in all likelihood, dancing. Bees are wasps. This is not a loose metaphor or a taxonomic technicality, it is a literal evolutionary fact. Bees descended from a group of predatory wasps in the family Crabronidae, and the transition from wasp to bee involved a single pivotal dietary shift. Instead of hunting insects and spiders to provision their larvae, a population of these wasps began provisioning with pollen. The reasons for this shift are debated. One hypothesis suggests it began with wasps who were already visiting flowers to hunt the insects that fed on them accidentally, accumulating pollen on their bodies and inadvertently provisioning their larvae with a mixed diet. Over generations, those larvae that could survive and thrive on pollen survived better, and the behavior reinforced itself. The anatomical differences that distinguish bees from their wasp ancestors, the branched pollen-collecting body hairs, the specialized leg structures, evolved in response to this dietary shift, accumulated over millions of generations of incremental selection. The hive, the dance, the democracy, all of it traces back to a wasp that at some point in the deep past landed on a flower and did not immediately leave. In the iconography of ancient Egypt, the bee held a position of unusual dignity. The hieroglyph for the bee, a small, carefully rendered symbol recognizable in tomb paintings and administrative records alike, was used as part of the royal titulary of the pharaohs of Lower Egypt from at least the first dynasty around 3100 BC, making it one of the oldest continuously used royal symbols in recorded history. Egyptian temples maintained apiaries. Honey was used in medicine, in ritual offerings, and in the preservation of the dead. The association between honey and immortality was direct and practical. Honey does not spoil, and the Egyptians knew this. Jars of honey placed in tombs were intended as provisions for the afterlife, a food so stable it could outlast the person who left it. Archaeologists have found honey in Egyptian tombs that is thousands of years old and under the right conditions of storage still chemically recognizable as honey. The bee in the Egyptian imagination was not merely a useful insect. It was a creature that produced a substance that time itself could not easily touch. In ancient Greek tradition, bees occupied a liminal position, creatures that moved between the world of the living and something beyond it. The nymphs who nursed the infant Zeus on Mount Ida were in some tellings described as bees or bee maidens. The Delphic oracle, whose pronouncements shaped the course of Greek political and military history for centuries, was associated with bees. The title Melissa, meaning bee, was given to the oracle's priestesses, and the gift of prophecy was sometimes described as a sweetness poured into the mouth by bees. The soul, in some Greek traditions, was thought to take the form of a bee, at death entering and exiting the body through the mouth, traveling between worlds as bees moved between the hive and the open air. These mythological associations are not arbitrary. They reflect an accurate observation. Bees emerge from enclosed darkness into light return, laden with what they have gathered from the world, and produce from it something that does not decay. For a culture thinking carefully about death and what might lie beyond it, the bee was a reasonable symbol to reach for. Among the most celebrated artifacts of the Minoan civilization, the Bronze Age culture centered on the island of Crete, flourishing approximately 1800 to 1500 BCE, is a small gold pendant known as the bee pendant of Malea. Discovered in a burial site in the 1930s and now held in the Heraclean Archaeological Museum. The pendant depicts two bees facing each other, their bodies forming a symmetrical composition around a central honeycomb cell, their wings spread and joined above in an elegant arc. A granulated gold sphere hangs below a smaller sphere above. The craftsmanship is extraordinary for its period. The granulation technique, which involves fusing tiny spheres of gold to a surface, requires a level of metallurgical control that challenges modern jewelers attempting to replicate it. The pendant was almost certainly a prestige object, worn or displayed by someone of significant social standing. That a civilization should choose the bee, its symmetry, its cooperation, its golden product, as the subject of its finest jewelry, tells you something about how deeply these creatures had embedded themselves in the human imagination, long before anyone had the tools to explain what they actually were. Mead fermented honey, dissolved in water, is the oldest alcoholic beverage for which there is direct archaeological evidence. Residue analysis of pottery fragments from a site in northern China, dated to around 7000 BCE, revealed the chemical signatures of honey, rice, and fruit fermentation, suggesting a mixed fermented drink that included honey as a primary ingredient. In Europe, dedicated mead production appears in the archaeological record somewhat later, but spreads widely, finding its way into the ritual and social life of cultures, from Scandinavia to the Mediterranean. In Norse mythology, mead was the drink of the gods, the mead of poetry brewed from the blood of the wise being kvasir, mixed with honey, conferred the gift of eloquence and verse on whoever drank it. In Welsh tradition, mead halls were the centers of social and political life. In Sanskrit texts, madhu, the word for both honey and mead, appears among the oldest layers of the language. Across cultures that had no contact with one another, the same discovery seems to have been made independently, that honey left in water becomes something more than honey, something that loosens the tongue, warms the chest, and apparently reaches the gods. In medieval Europe, bees occupied an unusual legal position, neither fully wild animal nor fully owned property, but something in between that required its own body of law to manage. The Brehon laws of early Ireland devoted considerable attention to bees, specifying in careful detail who owned the honey from swarms that crossed property boundaries, how compensation should be calculated when a neighbor's bees stung someone's livestock, and what obligations a beekeeper had to neighbors whose flowers his bees visited. Welsh law codes set the value of a hive of bees at a fixed price, greater than the value of many domestic animals, and specified the precise procedures for claiming ownership of a swarm in flight. In parts of Germany, a beekeeper who followed a swarm across a neighbor's land without permission was legally protected provided he kept the swarm in sight and announced his presence. The elaborateness of these legal frameworks reflects something important. Bees were economically significant enough, and their behavior ambiguous enough, in terms of ownership and responsibility, that societies found it necessary to think carefully about how to integrate them into human legal and moral structures. They were not livestock. They were not wildlife. They were bees, and they required their own category. Across rural communities in Britain, Ireland, France, Germany, and parts of New England, there existed, until surprisingly recently, a folk practice known as telling the bees. When a significant event occurred in a household, most commonly a death but also a birth or a marriage, a family member would go to the hives and formally inform the bees of what had happened. The announcement was made in a low voice, often accompanied by a knock on the hive. In some traditions, the hives were draped in black cloth to mark mourning. If the bees were not told, it was believed they would leave, stop producing honey, or die. The practice has been documented in literary and ethnographic sources from at least the 17th century and was recorded as a living custom in parts of rural England into the 20th. Its origins are genuinely obscure. Historians debate whether it reflects ancient pre-Christian belief systems in which bees were thought to carry messages between the living and the dead, or whether it developed more organically from the practical reality that hives require attentive management during disruptions to household routine. What is beyond debate is that for centuries, people felt an obligation to their bees that extended into the emotional and ceremonial dimensions of human life. The bees were not kept. They were, in some meaningful sense, included. William Butler Yeats, writing in 1888, imagined a place of peace and stillness, where he would hear lake water lapping with low sounds by the shore and live alone in the bee-loud glade. The phrase has become one of the more durable images in English poetry, and it names something that is genuinely harder to find than it once was. Entomologists studying historical records museum collections and long-term field surveys have documented dramatic declines in wild bee populations across Europe and North America over the past several decades, with some studies suggesting losses of 50% or more in species richness in heavily agricultural regions. The sound of a bee-loud glade, the layered hum of multiple species working a diverse wildflower meadow, is in many parts of the world a quieter sound than it was in Yeats's time, and quieter still than it was a century before him. Scientists debate the relative contributions of habitat loss, pesticide exposure, introduced pathogens, and climate-driven phenological mismatch to these declines, and the picture varies significantly by region and species group. What is consistent across the data is the direction of change. The glade is still there. It is simply less loud. Honey is one of the few organic substances that under the right conditions can survive for thousands of years without significant degradation. Its low moisture content, high sugar concentration, acidic pH, and the presence of naturally occurring hydrogen peroxide create an environment in which most microorganisms cannot survive. Archaeologists have found honey in sealed ceramic vessels in Egyptian tombs dating to over 3,000 years ago, and in at least some cases, the honey was reported to be still recognizable as honey in texture and composition, though its edibility was not tested systematically in most instances. Pollen grains preserved in ancient honey samples have been used to reconstruct the botanical environments of past landscapes, providing ecological data that would otherwise be unrecoverable. Honey residues in pottery sherds have helped trace ancient trade routes, since the pollen composition of honey reflects the flora of the region where it was produced. The bee's product outlasts the bee, outlasts the beekeeper, outlasts the civilization that stored it. There is something in that persistence that sits quietly at the edge of thought, the hive's labor sealed in clay, waiting in the dark for someone to find it centuries later and recognize it still. Napoleon Bonaparte chose the bee as his imperial symbol, a deliberate rejection of the Bourbon fleur-de-lis, and an equally deliberate claim to a deeper French antiquity. The choice was grounded in archaeology. Golden bee-shaped ornaments had been discovered in the tomb of Childeric I, the 5th century Frankish king, whose grave goods were unearthed in 1653, and Napoleon appropriated these as emblems of a pre-feudal, pre-aristocratic sovereignty rooted in labor organization and collective purpose. Bees were embroidered onto his coronation robes, cast in bronze on imperial furniture, and woven into the iconography of the Napoleonic state at every level. The symbolism was pointed. The bee works, the bee builds. The bee serves the collective without ego or leisure. Whether Napoleon's own administration embodied these values is a question history has answered with considerable ambiguity. But the choice of symbol was not arbitrary, and its logic was coherent. The hive had always suggested to those who watched it a model of productive order, an image of what human society might look like if it were organized around function rather than inheritance. Napoleon was not the first ruler to reach for that image, and he was not the last. The quote is, "Everywhere. If the bee disappeared off the surface of the globe, man would have only 4 years left to live." It is attributed to Albert Einstein in countless articles, speeches, and environmental campaigns. Einstein never said it. There is no credible source connecting the statement to him, no published text, no correspondence, no recorded speech. The earliest documented appearance of the quote, in its current form, dates to the 1990s, when it began circulating in European beekeeping newsletters, already attributed to Einstein without citation. Despite repeated corrections by historians and Einstein scholars, the quote continues to be reproduced and cited. What the misattribution reveals is not primarily about bees. It is about the human desire to anchor urgent environmental arguments to authoritative voices. The claim itself that human agriculture would collapse within years of a total bee disappearance is a significant exaggeration of what the science actually shows, which is serious but more complex. Bees are essential to the pollination of many crops and wild plants, and their decline has measurable consequences. Those consequences do not require a famous physicist's endorsement to be worth taking seriously. They are serious enough on their own evidence. Long before European honeybees arrived in the Americas, the Maya of the Yucatan Peninsula had developed a sophisticated relationship with a native stingless bee called Melipona beechi, known in Mayan as Xunan Cab, roughly translatable as the royal lady. Meliponiculture, the keeping of stingless bees, was practiced by the Maya for at least 3,000 years, and the bees held a sacred status that was embedded in religious practice, cosmology, and agricultural ceremony. Musan Cab, the Mayan god of bees and honey, was depicted in the codices with bee characteristics, and honey was used in ritual offerings in the preparation of a ceremonial drink called balshe, and in medicine. The hive's hollowed logs, sealed with clay and bark at both ends, were kept in dedicated structures and passed down through family lines as valued inheritance. Colonial disruption, land conversion, and the introduction of African honeybees severely reduced Melipona populations and the traditional knowledge associated with their keeping. Efforts to revive meliponiculture in the Yucatan have gained momentum in recent decades, driven by both ecological concern and cultural recovery. The Xunan Cab is still there. The knowledge of how to keep her is being carefully reassembled. In the winter of 2006 to 2007, beekeepers across the United States began reporting a phenomenon that did not fit any previously documented pattern of colony loss. Hives found nearly empty, the adult worker population vanished, with honey and brood still present and the queen still alive, but attended by only a handful of bees. No dead workers were found in or near the hive. They had simply gone and not returned. The phenomenon was named colony collapse disorder, and its cause remains, despite nearly 2 decades of research, incompletely explained. The scientific consensus that has emerged points not to a single cause, but to a convergence of stressors: the parasitic mite, Varroa destructor, which weakens bees and vectors multiple viruses; exposure to a class of pesticides called neonicotinoids, which impair navigation and immune function; nutritional deficiency from monoculture landscapes; and pathogen loads from globally traded bee populations. No single factor reproduces the syndrome reliably in isolation. The interaction among them, in combinations and intensities that vary by region and season, appears to tip colonies past a threshold from which recovery is not possible. The hive empties. The bees do not return. Where they go remains in the strictest sense unknown. The economic value of insect pollination to global agriculture has been estimated in various studies at figures ranging from hundreds of billions to over $1 trillion annually. Numbers so large they become difficult to hold in any concrete way. A more graspable version: roughly 1 in every 3 bites of food a person in a developed country eats exists because a bee or another pollinating insect transferred pollen from one flower to another at the right moment in the growing season. Almonds in California provide one of the starkest illustrations the almond industry requires. the pollination services of approximately 2 million managed honeybee colonies each year, trucked in from across the country in what amounts to the largest planned pollination event in human history. Without this managed pollination, almond production would essentially cease. The same dependency at varying degrees of severity applies to blueberries, cherries, cucumbers, squash, and dozens of other crops. What bees do in the course of their normal foraging lives, collecting food for themselves and their colony produces, as an incidental consequence, the reproductive success of the plants they visit, which underpins the food security of several billion people. The bees are not doing it for us. They are simply doing it, and we have arranged our food systems around the fact that they do. The honeybee, the one most people picture, the one that lives in hives and makes the honey found in stores, represents a single species in a world of approximately 20,000. The vast majority of those other species are solitary: no queen, no colony, no honey surplus, no waggle dance. A female mason bee, for instance, builds her nest alone in a hollow stem or a small cavity in a wall. Provisions each cell with a ball of pollen and nectar, lays a single egg, seals the cell, and moves on. She will never meet her offspring. A mining bee excavates a burrow in bare soil, works alone for a few weeks, and dies before her young emerge. These solitary bees are in many ecosystems more effective pollinators per individual than honeybees. Their foraging behavior is less selective. Their pollen transfer more complete, and their activity periods often extend into cooler temperatures and earlier morning hours than honeybee foragers typically manage. Their decline, which has been steeper in many regions than that of managed honeybees, receives less public attention because they produce nothing humans directly harvest. They simply pollinate quietly without advertisement, and ask nothing in return except that there be flowers and somewhere small and undisturbed to build a home. In 1984-33, honeybees traveled aboard the Space Shuttle Challenger as part of a student-designed experiment to study whether bees could construct comb in microgravity. They could with some modifications. The comb they built in weightless conditions showed irregularities that ground-based comb does not. Cells that wandered from the precise hexagonal geometry maintained in normal gravity, structural variations that suggested the bees were using gravity as one of several reference cues during construction. The bees also flew differently, moving in ways described by researchers as less coordinated than normal flight, though they adapted over the course of the experiment. Most died before the shuttle returned. Their food supply was insufficient for the duration, but the experiment established that bee behavior is not entirely dependent on Earth's gravitational field, while also demonstrating that gravity is a more significant component of normal hive construction than had been previously assumed. The bees did not know they were in space. They simply found themselves in a place where some of their usual reference points were missing, and they built anyway as best they could with what remained available to them. The waggle dance was known to beekeepers for centuries as a curious bee behavior, interesting, perhaps, but not understood as communication. It was the Austrian ethologist Karl von Frisch who, through decades of meticulous experimental work, beginning in the 1920s and culminating in publications in the 1940s and 1950s, decoded it. Von Frisch trained bees to visit feeding stations at known distances and directions from the hive, then filmed and analyzed the dances of returning foragers, correlating dance parameters with the locations he had specified. The result was a complete cipher duration of the waggle run, mapped to distance angle, mapped to direction relative to the sun dance, vigor mapped to quality. In 1973, von Frisch received the Nobel Prize in Physiology or Medicine, shared with Nikolaus Tinbergen and Konrad Lorenz, for this and other contributions to the study of animal behavior. The Nobel Committee's decision to recognize the study of animal communication and instinct was itself significant. It established that understanding how other creatures perceive and navigate their world was a scientific question worthy of the highest recognition, not a peripheral curiosity. Von Frisch spent a lifetime listening to what the bees were saying. The prize was in part for having had the patience to hear it. The relationship between humans and bees did not begin with domestication in any conventional sense. Bees were never bred for docility or dependence in the way that dogs or cattle were. What happened instead was a gradual formalization of a relationship that began with honey hunting. The earliest direct evidence of humans deliberately harvesting honey comes from rock art. Paintings in sites across Spain, India, and southern Africa depict figures climbing ropes or ladders to reach wild bee nests in images dated to between 8,000 and 15,000 years ago. Deliberate hive-keeping, providing artificial cavities for colonies to occupy in locations convenient for humans, appears in the archaeological record in Egypt and the Middle East around 10,000 years ago. The clay cylinder hives of ancient Egypt were designed not to change the bee but to position it. The bee was never domesticated in the biological sense. It remains genetically a wild animal. What changed across 10 millennia was not the bee, but the human learning incrementally how to make a place for the bee in the human world without asking it to become something other than what it is. For as long as people have watched bee colonies, they have found in them a mirror or a reproach for human social arrangements. Aristotle described the bee community as a model of natural hierarchy. Mandeville's 1714 satirical poem The Fable of the Bees used the hive to argue that private vice produces public benefit, a foundational text in the development of modern economic thought. Marxist writers saw in the hive an image of collective labor, systematically appropriated by an idle class. Feminist scholars of the 19th century noted that the hive is, in practical terms, a matriarchy. All the labor performed by females, the males present only briefly for reproductive purposes and expelled when no longer needed. Political philosophers across the spectrum have found in the bee colony the pattern they were already looking for: order, cooperation, hierarchy, equality, industry, sacrifice. The hive is generous in what it offers to the imagination. It contains enough complexity that almost any social vision can find its reflection there, and has been finding it for at least 2,500 years. What the bees make of this sustained human attention is not recorded. Honeybees sleep. This was formally confirmed by research in the 1980s and has been studied with increasing sophistication since. Sleep in bees meets the behavioral criteria used to define sleep across the animal kingdom. It is a state of reduced responsiveness to external stimuli. It occurs at predictable times. It is reversible, and it is subject to homeostatic regulation. Bees deprived of sleep show subsequent rebound sleep, sleeping longer and more deeply after deprivation, in exactly the pattern seen in sleep-deprived mammals. Forager bees sleep primarily at night when foraging is not possible, accumulating roughly 6 to 8 hours of rest across multiple shorter bouts rather than one continuous period. Younger bees whose tasks are confined to the interior of the hive sleep in less regular patterns, catching rest in the spaces between nursing duties throughout the day and night. The hive does not sleep all at once. It sleeps in shifts continuously active at the colony level, while individual bees take the rest they need. Sleep is not a vulnerability the colony exposes. It is a function it has quietly accommodated, distributing it across the population so that no moment is ever entirely unattended. A sleeping bee is recognizable if you know what to look for. The antennae normally held forward and slightly apart in alert readiness droop, falling toward the body, sometimes resting against the surface below. The muscles that hold the head upright relax, and the head tilts forward or to one side. The legs which grip the comb or a surface edge while the bee rests sometimes loosen slightly, and a deeply sleeping bee may hang by fewer legs than she would while active, or sway gently if the surface she rests on is disturbed. The abdomen, which moves with each breath in an active bee, stills almost completely. Forager bees, older and more deeply fatigued, often sleep outside the cluster alone on a comb surface or near the hive entrance. At some distance from the warmth and press of the colony's center, a bee sleeping alone on a comb surface antennae, down body stilled, is a small and quiet thing to observe. There is nothing in her posture that announces her as one of the world's most complex social animals. She is simply resting as everything alive must rest after the long work of the day. The hive entrance is never fully unguarded. Guard bees operate in shifts, and the shift system accommodates sleep without ever leaving the colony's threshold unattended. As one guard moves inward to rest, another takes her position not through any explicit handover, but through the same kind of local behavioral rule that governs so much of what the colony does. When a position is vacant and the chemical environment signals that guarding is needed, a bee fills it. At night, when flight activity outside the hive drops to near zero and the primary threats shift from robber bees and wasps to crawling predators and condensation management, the guard contingent adjusts fewer bees at the entrance, their vigil less intense but unbroken. The colony sleeps the way a city sleeps, most of it quieter, some of it still active, the essential functions continuing at reduced capacity through the dark hours, ready to resume full operation when the light returns and the temperature rises and the flowers open again for another day. The entrance holds. It always holds. Inside a honeybee hive at 3 in the morning, the temperature in the brood nest remains what it was at noon, 35 degrees Celsius, maintained by the slow metabolic heat of the sleeping cluster. The comb holds its honey in sealed cells capped with wax, the sugars stable and unchanged by the hour. A few bees move through the dark interior, not many, and not quickly, performing the minimal maintenance that cannot wait for morning, adjusting the cluster's position, tending brood cells at the margins of the nest, removing a bee who died in the night. The fanning that ventilates the hive during the day has slowed to almost nothing. The colony's metabolic rate is low enough that the night air is sufficient. The queen is still. She sleeps in brief intervals, surrounded by a reduced retinue that grooms her lightly and then rests beside her. The waggle dance floor is empty. No scout is advertising anything. The hive's great deliberative machinery, its democracy, its intelligence, its layered communication systems, is suspended temporarily in the ordinary silence of a living thing at rest. The comb does not change. The honey does not move. The whole enterprise pauses in the dark in the warmth breathing. A bee does not breathe the way you breathe. There are no lungs, no diaphragm, no chest rising and falling with each cycle. Instead, oxygen reaches the bee's cells through a network of fine tubes, called tracheae air passages, that open to the outside through small pores, called spiracles, along the sides of the thorax and abdomen, branching inward in progressively finer divisions until they terminate in fluid-filled tips directly adjacent to the tissues they supply. Gas exchange happens at those tips without any circulatory intermediary. At rest, the spiracles open and close in cycles, sometimes with long pauses between openings, a pattern called discontinuous gas exchange, which reduces water loss through the breathing apparatus. In a sleeping bee, these cycles lengthen. The abdomen moves almost imperceptibly. The bee is exchanging gas with the air around her in a process so quiet and so internal that it leaves almost no external trace. She breathes in the dark, without breathing a distinction that matters only to the one watching, and dissolves entirely in the watching. As a honeybee colony settles into its overnight rest, the metabolic heat generated by the cluster decreases slightly from its daytime peak. The brood nest temperature is defended at all costs. The developing larvae require their narrow thermal band, regardless of the hour. But the outer zones of the cluster, where sleeping workers huddle in dense layers, run cooler than during peak activity. The individual bee at the cluster's surface, pressed against her neighbors, radiates warmth inward and absorbs the cold of the surrounding air. Her own body temperature is not fixed the way a mammal's is. She is not fully endothermic. She tolerates a degree of cooling that a mammal could not, her metabolism slowing with the temperature, her sleep deepening as she cools. In the morning, as ambient light increases and external temperature begins to rise, the cluster stirs before any individual bee makes a conscious decision to stir. The warming is collective. The waking is collective. The colony emerges from sleep the way it does everything else, not one bee at a time, but as a single distributed system, responding to the same signal simultaneously from the inside out. Even the queen sleeps. Her rest is briefer and more interrupted than that of the workers around her. Her laying schedule, which during peak season may involve 2,000 eggs in a single day, does not pause entirely at night, though it slows. She sleeps in short intervals between bouts of laying, surrounded by her retinue, which also rests in shifts so that she is never entirely unattended. Her sleep posture is the same as any bee's antennae, softened head tilted, the body's small movements stilling, but she sleeps at the warm center of the cluster, held in the collective heat of 50,000 bodies arranged without plan or instruction around her. She does not know she is at the center. She does not know there is a center. She lays and rests and lays again. The chemical signals her body produces, continuing, even in sleep, diffusing outward through the retinue, through trophallaxis, through the hive's chemical conversation that does not entirely stop, even when its participants are no longer fully awake. The colony's deepest governance continues in the dark, in the warmth, in the long quiet between midnight and dawn. A honeybee worker who survives long enough to become a forager, which requires surviving the first 2 to 3 weeks of adult life, as a nurse, wax producer and guard, will typically forage for the final 2 to 3 weeks of her life. During that time, she may make 10 or more foraging flights per day, each one covering several kilometers of terrain, each one requiring sustained high-frequency wing beats in varying weather conditions against wind, through turbulence loaded with nectar or pollen or water on the return journey. The cumulative physical cost is visible. Older foragers show measurably worn wings, the membrane thinning toward transparency at the tips and trailing edges. Small tears and notches accumulating where the wing surface has been stressed past its resilience. The wing cannot repair itself. Each flight wears it a little more. A very old forager near the end of her life carries wings that are tattered, things still functional, barely still beating at the same frequency they always did, but diminished by the accumulated evidence of everything they have carried her through. She does not slow. She does not stop early. She flies until she cannot, and then she does not come back. Honeybee workers do not typically die in the hive. They die outside it. A forager who has reached the end of her life, her wings worn, her fat reserves depleted, her flight muscles approaching failure, leaves the hive on what will be her final trip and does not return. Whether this is a voluntary departure or simply a foraging flight from which she lacks the resources to return is a question that cannot be answered with certainty, because no one has yet found a way to ask a bee what she intends when she crosses the threshold for the last time. What is known is that hive interiors do not accumulate large numbers of dead workers. They die elsewhere in the field, in the grass, under a flower, somewhere along a route. They have flown dozens or hundreds of times before. The colony does not mark the departure. There is no ceremony and no searching. The chemical conversation continues without her voice in it. The dance floor fills with other dancers. The brood she nursed weeks ago emerges into a hive that has already, without hesitation or grief, made room for what comes next. She goes out into the light and the warmth and the smell of whatever is flowering, and she does not come back, and the hive endures. Somewhere in the last minutes before dawn, the air temperature crosses a threshold. It is not a large crossing, a degree perhaps, or less, but the cluster feels it. Not any individual bee feels it exactly. The sensation is distributed, registered at the surface of the cluster, where hundreds of bees are in contact with the cooling and then the warming air simultaneously. their bodies responsive to the change, before any single one of them has done anything that could be called waking. And then slowly, the cluster begins to loosen. The bees at the interior begin to move. The metabolic rate rises. Heat production increases. The first light reaching the hive entrance is not yet enough to fly by. The foragers know this, though no one told them, and so they wait, warming themselves and the brood and the air of the hive with the patient, purposeful heat of a community that has been doing this longer than our species has existed. Outside, the flowers that closed last night are beginning to open. The pollen that was tucked away in darkness is becoming available again. The world is incrementally becoming the world again. And when the light is sufficient, not before, not after, but precisely when it is sufficient, the first forager crosses the threshold, spreads her worn or unworn wings, and lifts into the morning air. The hive endures. It has always endured. It will go on doing so long after this night's quiet has been forgotten, carrying its democracy and its memory and its warmth forward into another day, as it has carried them forward through 100 million years of mornings just like this one, and just like none other.